I'm investigating EOE. Have you heard about eosinophilic esophagitis? So I'm trying to understand what happens in EOE. Why does eosinophils get into the esophagus and start inflammation in the esophagus? And our esophagus consists of a small tube, and the most outer layer is epithelial cells. So I'm trying to see what happens with the epithelial cells, because they are secreting molecules called cytokines, and they are attracting the eosinophils over there. So, and those epithelial cells, they are like our barrier, so they should prevent from molecules from food to get into the tissues, because it just need to go to the digestive system. So we try to understand what is the genetics and the molecules that are causing this impaired barrier and the release of molecules that attract the eosinophils and other immune cells to the esophagus and cause inflammation.
Cool.
Some of the kids with EOE have it in multiple generations of the family. So like you, and then mom, dad, and then grandma, grandpa, and then some of our kids have it only in just one person. Only one person in their entire family. And so that's one thing that Tetsuo looks at, is the families that have it in multiple generations, because that's very interesting to us. That's genetic. If you can prove it in many generations and then have it in all these different family members, that's considered a possibility for genetics. And that's what he looks at. So we map big family trees. Everything is de-identified, so you'll never know whose is whose. So one of the things that we get from you is a biopsy, right? Have you ever seen a biopsy? The ones that they take from you when you get a scope? No. So you see that little guy right there? That's a biopsy.
So tiny.
Yep, that's a little baby. And then what they do in pathology, which is one of the reasons you get a scope, is they put it in wax and they cut it and they put it on a slide. So there it is in wax. There's a biopsy. You can see it. This one's already been cut, so it's a little easier to see, right? And then we put it on a slide and we stain it. So there's two pieces that have been cut and stained. And then they look under a microscope, which we're going to look at a little later. We've got some slides to look at. I'm going to show you. I have my awesome big picture here. Let's see. That's a biopsy. This is what we look at when we're looking at your eosinophils. That's an eosinophil there. Yep.
So right in here, the big pink ones.
Yep. Yeah,
So that
there's an
big
eosinophil.
picture is that little square under a microscope.
Yep. And this is what they count. So when they're counting, you know, when you get that count of eosinophils to see if you're active disease or not, this is what they're looking at. They're looking to see if they can count a certain amount within a certain field. So this is bigger than what they count on. They count on a 40x. This is a 20x picture, because you can't get everything into a 40x. But yeah, so they'll take it's almost like half of this, and they'll say, can I see 15 in that field? And if they can see 15, then that's your count, and that's your active disease. You should have none. So healthy people would have none. 0, none.
In their esophagus.
In their esophagus, correct. It is in your blood, though. You're supposed to have eosinophils in your blood. And that's what we're looking for. We're looking for this, the big guy. So,
What's that other picture?
so the other picture is not, I'm missing a few words, but this is your digestive system. But if you think of your GI system, it's basically one big tube, all the way down. And you just take nutrients from that tube. Yep. Alright, so I'm going to show you a little bit of other things that we do. So we like to take that small little biopsy that I showed you, and we like to cut it up into smaller pieces and grow millions of cells from them. So we put them on a plate. This is Dr. Rockman did this, and this is a barrier that we've put here, right? Think of it as a wound, right? So like if you cut yourself or something, and these cells on either side have been hanging out there until you remove that barrier, and then we wanted to see what would happen with those cells and if they would move to overcome that barrier, and they actually do. So there they go.
That's cool.
Yep. And this is over a whole evening. So like took a picture A time lapse, yeah, every minute over the night. And that's how long it took. And they're all color coded so that the computer can track each cell and see how far they move and what they do.
So eosinophils usually aren't in the esophagus, but they come in when there's either a wound or when there's an infection. So this mimics that, and we see how they migrate, how they move.
Yep, it's a great explanation.
So that eventually we can prevent it.
Yes.
Correct.
So we're trying to learn about how it's working normally and in tissue from the patients with eosinophilic esophagitis to see if there's differences.
It also lets you see the movement of cells, because if you think about it, like you see this picture and it looks so like stationary, so still. But your cells are moving, and they're there, and they're constantly going through cell cycles. So it's not just that they sit there and they're very stationary. This is fixated on a slide with the pictures that you see. So that kind of helps you see the movement, like your cells really do move. And there's, the eosinophils move through the tissue as well. That's why you see them within the tissue instead of on the outside, because they've been able to move between cells to get into the tissue. Yes?
What does CCED mean?
I'm gonna let Julie take that one.
So CCD is Cincinnati Center for Eosinophilic Disorders. So that's the name of our clinic, and that's where you see Dr. Putnam, right? And so we just call it CCED for short, instead of Cincinnati Center for Eosinophilic Disorders. It takes too long to say.
It does take too long.
Yeah, we're using a lot of abbreviations. So EOE that we keep mentioning is eosinophilic esophagitis. That means the eosinophils that are all moving in are in the esophagus instead of in the stomach, which would be eosinophilic gastritis, or in the colon, which would be eosinophilic colitis,
Which Johnnie has all of.
which we
You have
call
all
EG or
of
EC.
those.
We have an acronym for all of those though, and that's EGID. So
Eosinophilic gastrointestinal disorders.
yep, and that encompasses all of them. EOE, EG, EC. So if you have all of them, you have EGID.
It's kind of our umbrella term.
Yeah, it's the big term.
Cool.
So, yeah. So other things that we get from you when we ask for wonderful, beautiful samples is blood. So one of the things we get is we get a red top and a green top. The red top is processed immediately, so I don't have one to show you. The green top, we process the plasma off of it. Do you see the clear layer there?
Yeah.
Yep, and then there's a white kind of pellet sitting on top of the red blood cells. Do you see that?
Yep.
The white? Okay.
Kind of looks like what's inside that.
And that's a white blood cell pellet, and that's where we get your DNA. That's the cleanest version of your DNA. We can also do saliva, so if we're really interested in your family and the genetics, we will call the family and we will send you guys spit kits, and we'll say, before you brush your teeth in the morning, I need you to spit for me. And then that would be another way of getting DNA without having to do a blood draw, because those hurt a lot. All right. So I think we're time for our experiment.
That reminds me of one time when I was in the hospital for a week. I had to get poked every morning.
You get poked at, oh, it's a lot of poking.
No. That's terrible.
Yeah, all right, so we're gonna have a little bit of fun here. Come on down here.
What's with the ice?
This is dry ice. Do you know what dry ice is?
Mist.
What? No, it's not mist. Do you see the pellets? Those are actual dry ice. You can't touch them. They're very cold. They'll burn you. They're so cold that they would actually burn you. That's why I'm wearing a glove. But we're gonna see something happen when I put it in the water.
Oh!
It's
Explosion! Sleep.
Yep. So that's just soap and
Does that
water
burn
and
me?
dry ice.
Oh.
You can also color it if you so choose. You can touch the bubbles. Go for it.
That would be awesome
It's just
for our
soapy water.
volcanoes.
Right?
Cool.
It's just soapy water. It's like washing your hands, but fun. Ah.
You might actually wash them if it was like this every time.
Just, see, now they look at it, right?
Yeah.
Look, they missed.
Braided. For instance, if you were harvesting cells, you could freeze them very quickly in liquid nitrogen and put them in long-term storage. If you're harvesting things from animals, usually you would put it in a tube and then pop it into liquid nitrogen so that the proteins don't change, because once something is harvested very quickly, there's proteins called proteinases that'll go, and other enzymes that'll go through and just chew up everything. So if you really want to know what's going on, you kind of have to like capture it at that one moment in time. And the liquid nitrogen is so cold that it helps us do that very, very quickly. Some other samples don't need that to that extent, so we just put them on ice until we get the window going. But for the really important stuff, you're dealing with enzymes, the molecular reactions, all the nitty-gritty stuff we don't see. Put in liquid nitrogen. Yes. Yes. Sorry.
What made you study us?
Oh. Well, I'm actually a medical writer in the division, so I do a lot of benchwork science first, and then I help with communicating science ideas because I think it's really important for everybody to learn more. But one of the main reasons here that we do eosinophilic esophagitis, we do the other eosinophilic gastrointestinal disorders, is because originally Dr. Wackenberg a long time ago was looking at the eosinophil. It's a white blood cell and investigating what it does. And that naturally led to some things that he was seeing in clinic about all of these unexplained cases of esophagitis. They just couldn't figure out why or what or how to treat these esophagitis cases that weren't responding to inhibiting acids. Because a lot of esophagitis is caused by acid from your stomach coming up in reflux. But that's not the case. And these were difficult to diagnose, and that presented an intriguing, situation that you just had to get to the bottom of. And I'm glad that we did. So now
we can help more kids, right?
So this is what we consider normal control. So this is someone that doesn't have an EGID disorder. And they're usually here for like motility or clean out, and they graciously give to research because they're going through a scope already, even though they don't have the disease. We value them very highly because in order to prove science and prove something in your population, you have to be able to prove it against a healthy person that doesn't have the disorder. And it's very important for us to have a normal control to base something against. So what I wanted to show you guys with this is like a baseline for what this looks like. Maybe. There it is.
That's blank.
Maybe not. There it is.
I
Alright.
see lines in it.
You do. So those are cells. So that's a basal layer, that top layer here. This is what's called a papillae. You'll see a lot of pink in the papillae, but those are red blood cells.
What's the bottom bit called?
That is part of the basal layer. Just a little bit different of an area. So those are red blood cells in the papillae, right there. You won't see any eosinophils in here because, again, this person doesn't have any eosinophils, or they shouldn't, because they're what we call a normal healthy patient. Just happens to be getting a scope. Those purple lines that you see are also just from folding from the wax. So with that one in mind, I'll show you our next slide. And this is someone that has like a lower count of eosinophils, so just over active is what we would call this person.
They have like right around 15 or...
Yeah, 15 to 20. Just over that active mark, right? And so you can already kind of see a little bit of a difference, right? So it's kind of a bigger layer, it's a lot darker of a purple.
Oh, there you go. What's with the weird yellowish red?
So those again are what's called papillae, and they have the red blood cells in them.
Ew.
So those are normal. What's not normal is the pink that you see outside of it. So just looking at it now, because I'm so used to looking at eosinophils, I can already tell you there's an eosinophil here, here, and here. And there's one over here. That might not be an eosinophil. We'll look in just a moment, because we're still on a 20x.
That one here and
Or on a
here and
10x.
here and here.
And we're gonna move down to our 40x. Let's see if the 40x can do it here.
So tiny.
My 40x doesn't like to do what it needs to do. Yeah, it is an eosinophil because there's a nucleus inside of it right there. And then there's most of them don't look like this. I don't know why this slide always does this to me. But yeah, so that's someone that has a lower count. And then let's go up to the range of like 60 eosinophils. So again, a normal person has 0, and this just has like 20, and it's already like a difference, right? And now we're looking at someone that has 60. So again, a lot darker of a purple. You can see a lot more red blood cells. So those are those big pockets of pink that you're seeing. And then right here is another cell layer. This is called lamina propria. The higher your eosinophil counts, the more fragile that tissue becomes, that we've noticed. And then we've been able to publish on, actually, because you have so much degradation from the eosinophils moving within the cells. It's not very compact. So normally your cells are very, very tight, right? So your cells are really tight, and they talk to each other, and they communicate with each other, and that's how they're supposed to be. When you have eosinophils and other cells that are moving through there that they normally shouldn't be there, your cells kind of lengthen out, and they don't really touch anymore, and they don't talk to each other, and that's when you get really fragile tissue, because it rips very easily open, because there's nothing to hold on to. Those cells can't hold on to each other. And so we get more cell layers from other cell layers that normally we wouldn't, because it's so fragile.
Can't hold my finger still.
So, and that'll be really apparent when I show you the one next that has over 100 eosinophils. This guy just has about 60. So can you see all the pink dots right there?
So many.
So many. So that's an eosinophil. So there's like 1, 2, 3, 4, 5, 6.
7, 12, 13.
But not all of these are eosinophils. Can you show me the one that's not? Actually, there's several on here that are not eosinophils. And I'll tell you why. So that's the really hard part about eosinophils, or counting eosinophils. Oh, it's not going to do it for me. Sorry, this is really blurry. So this is pink, and it's got that purple nucleus, right? So if you look at my badge, that's an eosinophil. To remind ourselves, we have pink granules, and then we have the purple nucleus. So in this slide, you have the pink with the purple. This is just pink. That's not an eosinophil anymore. You can't count it as an eosinophil. It doesn't go into the count. So when you're counting, you have to make sure it has that nucleus in it, even if it's like morphine to go through.
That yellow thingy.
Mm-hmm. Yep. So then we're gonna take a look at our last slide, and this
What's with the paper in it?
That's my cheat sheet, in case I forget, or in case someone else has to show these and I'm not here. That way they know what they're looking at. It helps us remember.
Whoa, that's outside the thingy.
That's just from the staining. So can you already tell the difference with this slide? This person has over 100 eosinophils in a 40x field. What's so different?
Whoa, there's so many.
You can't see them from there. What are we looking at when we look at this like this far away?
So many dots.
There are a lot of dots. Those are all cells. But what we're looking at is the biopsy as a whole. So do you see missing pieces like that have been torn away?
Those white little hole things.
Yes. And so those have been torn away because, again, that tissue is so fragile. Those cells have nothing to hold on to each other. So when we zoom in a little bit more, you can kind of see all the white between all the cells. It looks completely different than that normal biopsy.
Eosinophil thingies.
Yep. And then there's that bridge of eosinophils right there. Those are all eosinophils. All through here, those are all eosinophils.
The bridge of eosinophils.
And the bridge of eosinophils.
It must be really hard to count.
It is, and we count by hand.
By hand?
Yep. So, again, this isn't really showing up very well, but those are all pink with purple inside of them. But yeah, we count them by hand. So I look through this scope here. So the camera is actually a little smaller than the actual scope. So what you're seeing on the screen is smaller. And then it varies off of scopes. So a pathologist down in the actual clinical pathology will may have a bit of a bigger or smaller scope than I do, so we'll be off by 1 or 2. And then Dr. Collins, who is our research pathologist, she is who normally looks over eosinophil counts from slides that get sent from other places. And her scope is, I think, 1 millimeter or 2 bigger than mine, just millimeters. And she and I will have off counts because that's how small these cells are. So just looking at the field in here will have a little bit of a difference. And it's good to know that so that you're not comparing and saying, well, you're wrong and I'm right. It's, oh, well, you have a different scope than I do. Okay, like, you know, if you're off by like a lot, then that's a different story. But if you're off by one or two.
You look at a field too, so do you always look at the same spot?
No.
So that could account for
Yep.
the
So
differences too.
when they go through your slides of clinical pathology has 6 pieces. We have 2 because we conserve, and because we try to conserve the tissue that we get. And so clinical will skim each of the 6 and take the highest count from each section, and then they will choose which section had the highest. They'll go back to that section, and they will recount it, and that is your peak eosinophil count per high power field 40x on that biopsy. And they have to do the different pieces because like you can split an eosinophil in half. Like that's how small. It's like a 10 microns, I think, is what we cut at. I think it's 10. So it's very small slivers of pieces of tissue, and that's what we cut and stain. So you can have like 2 extra eosinophils on a different section, even though it's the same biopsy or the same area.
Right.
So, yep,
And then I know Dr. Putnam looks at them too.
he does.
Could we help?
How could you help? That is a Julie Daisy question.
What?
Thanks. How can kids help?
Yeah.
You're helping us every day by when you do your scopes and you give samples to research, right? And another thing that you're doing is helping to build awareness about EOE because you're teaching other kids, right? About what your disease is and how it affects you. And so the more people in the world know about it, the more researchers will want to learn. Good?
So what we're going to look at here, so when researchers want to publish research, they will have a poster, and then they will also have a publication, right? So they go and they present their research, and people are able to look at it and ask questions, and then they publish it in a paper. So what this is showing is where an eosinophil comes from. So an eosinophil will come from the GMP, and it's called a progenitor cell. And there's one up there too, where you can see it makes the decision to either become like a neutrophil or if it becomes an eosinophil, and that's what we're looking at there. Do you see that?
Yeah.